So just a short introduction. So in my lab and my research team, I work with my students and my collaborators, and we work on projects related to freshwater conservation science and macroecology, which means we use a lot of data science approaches to analyze data sets that are publicly available. A lot of these data sets are collected by state agencies, federal agencies like the EPA, USGS, and various states. And they include things like fish monitoring data, climate data, stream flow data, and things like that. And we use these data sets to really investigate and examine the impacts of climate change, land use change, and other human stressors, like invasive species on native fish communities. And then we also do like the mapping of threats and like conservation priorities. So if you're interested, you can visit my pretty outdated website. I've got to sort it out, and I promise I will. Or you can just contact me through my email, xgiam@utk.edu. Right. So before I start, I would really like to kind of acknowledge my co-authors and collaborators on this work and the funding sources. So this project really comes from a research grant that I got from CCAST, and I'm really grateful for their support for this work, because without their support, it would be impossible to do this. And my collaborators and as well, like my students like Guido, and then Taylor Woods, who has since graduated, and everyone who has helped me in this project and worked together with this, especially Jacob LaFontaine, who is a hydrologist out at USGS in Georgia, and he was really involved in kind of working with me and Guido and really guiding Guido in generating a lot of these stream flow outputs that we use for our analysis and for our predictions. And then, I would like to thank everyone in the state agencies, federal agencies, TVA, and nonprofits for so generously contributing the data sets, because without the people who actually collect the data in the field, it would be impossible to do synthesis projects like that. So I'm really grateful for their collaborations. All right. So we know that there has been an acceleration of warming over the past 50 years or so, from the news that we have heard and from the science that's been done. And we know that this global climate change is driven by human activities. And the southeastern US is a region that has also seen accelerated warming during this kind of most recent 50-year period. So if you look at this particular graph, you can see that since about 1970s onwards, there's been a kind of upturn in warming temperatures, and then all the way up to this current decade. And based on the different scenarios, the different routes that we take and the different policy choices that we make regarding decarbonization or continuing through business as usual, global surface temperatures might increase by up to like 5 degrees relative to the last century. And in the Southeast, we have seen this change from 1990 to 2019. I collected weather data from the different counties in the southeastern US, and we see that per decade there's been up to like half a degree increase in just the mean summer temperature. So we are not immune from this warming trend. And it's a problem because we know that the southeastern US is a really important region for biodiversity, and it's not just important for North America, it's actually really important globally. So this paper by Dr. Duncan Elkins and his collaborators out at the University of Georgia, they kind of and the Tennessee Aquarium, they kind of did a mapping exercise of hotspots of threatened and endemic biodiversity, aquatic biodiversity in the US, and they concluded that the southeastern United States is not just a national or continental hotspot, but it is a global hotspot for aquatic biodiversity. But yet there is very little land here that is like formally protected. And in the analysis, they did a conservation score thing, mapping, and they found that the highest scoring, the most important watersheds that should be prioritized for conservation are primarily in the Middle Tennessee River and in the eastern mobile river systems. So it's really relevant. So the kind of stuff that we are trying to do in the Tennessee River Basin is really important because, you know, independent studies have come out and have really shown and exemplified that, you know, the fish fauna, the mussel fauna is really important. So the question I'm interested in is, how does climate change actually impact stream fish? So one thing that we often would study in terms of climate change is, think about how will future climate change impact stream fish or other communities. Like, for instance, can the biodiversity, can the biota survive in place? Would it get too warm, or would it get too dry for the various species to actually survive? And then if they have to move, can they actually keep pace with the pace of climate change to kind of access the habitat that is more thermally and flow suitable for them? The other question we can also ask, and I think this is asked less often than the future question, is we can ask, how has climate change in the recent past actually altered biotic communities? So instead of looking forward, we can also kind of look retrospectively and look for the imprint of climate change on biodiversity and biodiversity communities. So previous work that myself and my collaborators, so Liz is a postdoc who worked with me, and along with our collaborators, Julian Olden, Pablo Tedesco, and Albert Ruhi, we looked at how climate change and land use change interact to drive changes in fish communities globally. So we compile a lot of data globally, and you can see that a lot of data is actually focused in the temperate regions. We have very little data in the tropical regions, but Guido, my student, is kind of working on that right now. So hopefully we'll have more information on tropical systems. But even then, the previous work has been focused on entire fish communities as a whole, rather than just looking at native or looking at non-native fish responses. So we don't really know how climate change might actually affect kind of non-native fish responses over a large spatial scale. And understanding this is really important, right? Because, we know that invasive species or non-native species has the potential to become invasive, and if they do, they can have potentially negative impacts on the very biodiverse environments that we have in the Southeast, in the Tennessee River Basin and in Cumberland. And further, with climate change, we know that the streams are going to warm, the flow regimes are going to change. And in the Southeast, we've got a wave of kind of urbanization, suburbanization, where a lot of housing subdivisions are kind of opening up. They are taking over cropland, or they are taking over like private forests that the landowners decide to sell away because they want to retire. So a lot of new urban development. And all these stresses might open up new habitats for the non-native species to invade and to thrive and impact our native biodiversity. And so they are really understanding the impacts and understanding how non-native species might change in their distribution and how they might impact native species would be management priorities for state fish and wildlife agencies as well as federal agencies. So a model system, to really study non-native fish responses to climate change is, I propose the Tennessee and Cumberland River Basins. Part of the reason is, we are located here in the Tennessee River Basin, Knoxville. And so, it's a system that's really dear to me. We visit the streams, we work with local nonprofits, and I've got native fish now in my lab that we are kind of growing out and doing some temperature climate experiments on. So it's a system that's really close to me. So these two systems, they are actually two independent systems, right? One is the Tennessee River Basin are all the basins that is kind of shaded in pink that you can see here to the south. And then we've got the Cumberland River Basin, and they are made out of two sub-basins, Upper Cumberland and the Lower Cumberland. So for both basins, the flow of the river flows from the east, then it goes southwest a little bit, and then it flows up northwest. So both rivers, they do not join up with each other. They join up with the Ohio River, actually. So I think the Cumberland River, they join up with the Ohio River at a city called Harlan in Kentucky. And the Tennessee River also joins up with the Ohio River, but in another city called Paducah in Western Kentucky. So there are two closely related systems. Both are very, very diverse in fish biodiversity. Both have a lot of endemics and a lot of threatened species, a lot of SGCNs. Oh, yeah. And also one more thing is like both, both of these drainages, they span multiple states, right? So they've got, the Tennessee River Basin will originate in the mountains of North Carolina, in Northeast Tennessee, as well as in Virginia, and then it goes down into Tennessee, it goes past Georgia a little bit, it goes to Alabama, Mississippi, back into Tennessee, and then into Kentucky. Whereas for the Cumberland River, it goes from Kentucky and then down to Tennessee and then back to Kentucky again before it joins up to Ohio. So for both the management of both of these rivers would require kind of cooperation and communication between the different state agencies. So it's a region that is kind of undergoing warming, and also is projected to continue warming. So the projected warming by the end of this century can be, in the western part of the Tennessee drainage can be up to about 6 degrees Celsius. And also, right now, as we have seen from the previous slide, we have seen that the southeast are in the area that is like currently warming as well. And beyond warming, precipitation change actually drives changes in flow regime, right? Because of climate change, it's not just when you think about climate change, we often think about warming and whether species are thermally suitable for, they're physiologically suitable to withstand the new thermal regimes, but we don't really think about precipitation that much. And for fish, precipitation is really important because changes in precipitation is the main driver of how runoff changes and the timing of stream flows and the amount of flows and the seasonality of flows. So, when we started this project, I came across this paper or this report by Jacob LaFontaine, which kind of like piqued my interest in really using flow, model flow, to actually get a better handle on how flow regimes might affect freshwater fish. Because like previously, a lot of data on flow is based on just gauges that are placed in really large rivers, and the way that most hydrologists do flow modeling, when they project flow over a global area, they're using more direct statistical correlational models, whereas Jacob and his team out at USGS are using more theoretical, more process-based models. So I was really interested and really intrigued by the product they were putting out. So with this product, we could actually better understand how flow, in addition to climate warming, might affect fish. So the research question that I have is really, these three main questions for this project. How has climate change impacted non-native fish richness and abundance in the past 30 years? And then how will the distributions shift in the future? And before we look in the future, we also want to look at how distribution have shifted in the recent past, which is over the past 30 years. And for this research talk, just for the interest of time, we're just going to focus on the first two questions, right? So how has climate change impacted trends in non-native fish richness and abundance in the past 30 years? And how will non-native fish distributions shift in the future? So two different questions, two different strategies of answering these questions. So for the entire project, the main thing that we need to do is to actually compile fish community and occurrence data. So when we say fish community data, it means that a crew of people from a state agency or from TVA will go down to different streams in the summer. Usually they start in March and they end their field sampling regime in about early September or so, or late August, and they go to a stream and they aim to sample comprehensively the entire community. So their aim is to get an overall idea of what species are there and what are the relative abundances. So when you talk about fish community, it's non-targeted sampling. The crews are just going there and trying to figure out what is the community that exists in the particular stream. And when you talk about occurrence data, it's data that is kind of present in museums or in databases like GBIF, where researchers, usually taxonomists, they will go to a particular location. They might be looking for one or two species, but they may or may not collect all the species that they encountered. So when they deposit or record the species in the museum, what happens is that for a given location on a given day, we can't be quite sure if the 2 or 3 species they recorded comprises all of the species that were there at the same time. So there's a difference between community data and occurrence data, right? So to do this, we collaborated with a lot of partners from state and federal agencies, land trusts and nonprofits, and we asked them if they could very kindly kind of contribute that data, and a lot of them was super helpful, and we asked them a lot of questions, and we're so thankful for that. So we compiled. Main thing is we wanted to compile fish community data at sites, and some of the sites were repeatedly sampled. So in different countries, like in a lot of European countries, like in France or in Sweden, there's like yearly sampling of sites. Whereas in the US, there is repeated sampling, but the sampling is not, most of the time it's not done every year. It's done every 3 years, 5 years. So there's a rotation of sites that they go through. And part of it is just manpower. It's impossible to get, go through like 1,000 sites in a single summer. So the crews will rotate through, and every 5 years they will repeat the sampling. So those are kind of the data that we have. And then for occurrence data, we got the data from museums, we got data from GBIF as well as online databases. So to compile all this data, we had to process them, we have to check them and standardize them. There was a lot of things that needed to be cleaned because different data sources, they can name species differently. Some data sets use common names, and even common names might be different from one person to another. Some might be written in shorthand. So we had to figure a lot of these things out. A lot of even if they were scientific names, a lot of the names were misspelled, or a lot of the names, especially for old museum specimens, they were using old names that were, so when, say, a taxonomist decides to split a species, they don't go back to the museum and relabel that species. So that data set still has the old name. So we have to go through a lot of original literature reports, a lot of descriptions. And for that, I work with Ben Keck, who is the curator at the ichthyological collection here at UTK, and work together to figure out, to sort through some of these things. But to compile all these sites and occurrences, to clean the data, it really took a lot of time. But I'm so thankful that we managed to do it, because this is, I think it's a one-of-a-kind data set, because it has just over the Tennessee River Basin and Cumberland River Basin, we've got about 3,225 sites with community samples. That is a lot. And if you look at the density of points over here, so the orange points over here are occurrence data. So those are data sets where we are not sure if researchers went there and they recorded every species they saw. So we can't do any community analysis. But the dots that are black are dots that we are sure that, or at least it was communicated to us or communicated via the database, that it was non-targeted sampling, and everything that they encountered and they sampled were recorded. So with that, we've got 43,740 unique sites in these basins, and more than almost 500,000 fish records, individual fish records. So, going through this data set it took so long to clean, but I think it's super worth it, and I couldn't have done it without all these groups of people that I mentioned earlier. And it's really a big effort, and I would like to thank my previous grad student Taylor Woods, who started this process, and then really like to thank Guido Herrera, who took over from Taylor when she graduated, And he really ran with the project. Like he did a lot of the technical stuff and cleaning stuff, and you know, over the past year or so, both he and I, we have worked together and, you know, to try to really get this data set down to so that we can use it not just for this project but also many other projects that I can talk about later. Right. So besides fish, very importantly, we want to have stream flow data as well. So we talk about the PRSM data, and the PRMS data. So PRMS data is a data set or data model that is designed and produced by USGS hydrology staff, and it started in the 2000s. And we use different calibrations and parameterizations of this PRMS model to basically for different research questions. So for the research question that we have, we may need to use a different version. Like for instance, if we want to run species distribution modeling, like so SDMs, and we require continental scale data to train our models, because the non-native fish that occur in the Tennessee River Basin doesn't just occur in the basin, it occurs in the conterminous US. So we have to use the entire US as a training region. So for that, we have to use the version of the data that Jacob Lafontaine and his team has produced. And that data is forced with the gridded climate forcings that's produced by Moore. And then for the community level analysis, because it requires only current contemporary data, because we are looking at how fish communities have changed in the past 30 years, for this data set, we actually took the model, and Guido actually worked with Jacob to actually run the model using our own climate forcings that we derived from GridMap. And the reason why we did that is because GridMap data is observed and it's more up to date than the Moore data, firstly, and also secondly, the resolution is actually higher. But we can only do it because, you know, we don't need to have that future simulations for this. The next step we have to do, you know, is also a difficult computational step, because the PRMS data that I've shown here, it outputs runoff in cubic feet per second, so flow or runoff, at the scale of what we call a hydrological response unit, HRU. So HRU is based on the National Hydrographic the NHM database. And HRUs are typically larger than catchments. So when we analyze fish data, we usually analyze fish data at the scale of the catchment. So a catchment is like a land area that contributes flow to a given stream reach that can be anything from like 500 meters to 4 or 5 kilometers that is roughly homogeneous within itself. So if you look at this particular diagram over here, this is the Tennessee River Basin. All these different colors are hydrological response units that the PRMS flow outputs are in, so they're bigger. What we need is we need to downscale that PRMS data, you know, and no one has done it before, because they haven't needed to work with fish people who needed data down to the COM-ID scale. So we had to intersect the catchment. So the catchment boundaries are the boundaries, the pixelated boundaries in gray, and we had to intersect these boundaries with the HRUs. And then by doing that, we could assign the runoff to each COM-ID HRU intersection, and then later we aggregate the COM-IDs again to the COM-ID level. So by doing that, we can actually distribute flow to each COM-ID, and then using the NHD plus 2's package in R, we could accumulate the runoff down the stream network. So we actually did this for the Tennessee River Basin, and we tested it, and the runoff that is reported that accumulates down at the mouth of the Tennessee River at Paducah, Kentucky, is pretty close to the reported numbers that is observed in the USGS gauges. So we're pretty happy about that. And then for historical and future land cover data, again, for SDMs, we use land cover data from Seoul. So this is an older data set, but we had to use it because it was consistent with the underlying data that we use for the PRMS stream flow runs. But for the community level analysis, so we use like more up-to-date grid map. We also use more up-to-date land cover data from the USGS LCMAP program. And then for stream temperature data, this is interesting. So stream temperature data came from our group. So it came from my lab. So it's a project that my first postdoc, Matt Schroyer, so I had a great fortune of working with Matt. And Matt was my first postdoc when I started at UTK in 2017. And he had this crazy idea. It's like, what if I go trek around the entire like Tennessee and Cumberland River Basins and the surrounding basins and put loggers down to figure out what are the stream temperature regimes? I'm like, yes, Matt, you can do it, but it would take a lot of time and you'll be very tired. You'll be driving around all day long. You'll be hiking along all day long. And he did it. He put 154 loggers across 154 stream reaches across the Tennessee and Cumberland. And I wish I could show you the map. I totally forgot, but it was all over the place. So he was really on the road for like 90, I think he was on the road for 80 to 90 days, in the whole summer, just to get to the loggers, just to download data and just to make sure that they're still there. But anyway, we have this really rich data set thanks to Matt. And we've got water temperature recorded at 15-minute intervals across the entire system. So we use this data and we train random forest models. So Guido trained these random forest models with hyperparameter tuning using the Ranger package in R. And then we predict the stream temperature as a function of air temperature, as well as hydrographic variables like elevation, upstream catchment area, stream order, variables like land cover, etc. Things that would affect, that we think would affect stream temperature. And then we did spatial cross-validation to select the top machine learning, the top random forest models. And our top model actually managed to generate daily max water temperature at an R square of 0.91. So it's very, very good. And we're quite happy about that. So this is a picture of like, for instance, the predictions that we have for, I think this is 2021, May, June, July, August, and September. These are the daily max water temperature in all of the months. Okay, so you can see the darker colors means it's cooler, and if you in the Tennessee and Cumberland River, right here at the bottom right-hand corner, or the bottom southeast corner, that is the Blue Ridge area, the Blue Ridge Mountain. So it's higher elevation, and you can see that it's darker, showing that it's cooler, right? And you can see that based on the variable importance plot, the air temperature is the best predictor of has the greatest effect on water temperature. All right, so let's get to the research. So from here you can see that it took us so much time to actually get the data, check the data, because as you did so many data processing steps, you tend to make mistakes, so we had to check the data very rigorously. So finally, time to get to the analysis. I think this is the interesting part. So first question, we want to look at the recent climate change impacts on non-native richness and abundance. So basically, identify climate change related drivers of non-native trends over the past 30 years. So what we do is, like, out of all the sites that we have, we choose sites where community sampling was done by the same organization repeatedly through the years. So kind of giving us some kind of a time series. It's not going to be a perfect yearly time series like what you might get in France or in different parts of Europe, but it's still pretty good. So we subsetted data with a range of more than 10 years, with the last year in the most recent decade because of the recent climate change that I mentioned. And each of the sites must have at least 4 sampling years. And sampling must be done within March to August, right? So that it's in the season where, it's easier to actually catch fish. Most people go out from March all the way to August. So we kept that sampling period at that time. So in total, we found 444 unique sites across 433 common IDs or unique catchments. And the mean year range of our time series is 19.1. So they're pretty long. A lot of the time series, they span, 15 to 21, 16 to 21 years. So that's the IQR, which is the interquartile range, the 25th and 75th percentile. The mean number of samples that we have, sampling, we have about 6.3 sampling occasions per time series or per site. And the mean day of year range tells you what is on average, the range of sampling events from the earliest to the latest throughout the time series. So the range is about, sampling is typically carried out within a period of about 3 months in general across all the sampling occasions in the time series. So using the USGS NAS database features of Tennessee and the original species descriptions, we determine what fish records are native and what fish records are non-native at HUC2 and HUC6 levels. So HUC6 would be all the basins of different colors that you see over here. And I forgot to mention that we did this study only for the Tennessee River Basin because in the Tennessee River Basin, we had a lot more sites that had the adequate repeated sampling, whereas in the Cumberland, we couldn't get that many sites. So we decided to focus this part of the project on the Tennessee River Basin. Right, so we determine whether a fish record or fish observation within a community is non-native or not, and we define native or non-native based on 2 levels, HUC2 and HUC6 level, which means if a species is considered non-native at HUC2 level, it means that that species, no matter where it is, it's just not native to any sub-basin within the Tennessee River. So fishes like rainbow trout, red breast sunfish, yellow perch, grass carp, brown trout, etc., etc. All these species are considered as non-native because they are not native to anywhere within the Tennessee River Basin. There's another group of species that we categorize as non-native at HUC6 level, which means that some of these species, just a few, we have a few of them. These species are present in the Tennessee River Basin. They're native to some HUC6 basins. So for instance, they might be native to the Holston or French Broad River, but then they might be non-native in lower Tennessee. For instance, they might invade one of the lakes here, maybe Lake Kentucky, right? So in that case, for that particular site, they will be considered non-native because they're not native to that HUC6, although they're native within the larger Tennessee River Basin. So one example, as I said, as mentioned, brook trout is one example, is native to the Blue Ridge ecoregion, so it's very common in the over here in the French Broad Holston, in the Upper Tennessee Basin, in the Blue Ridge ecoregion. But then there are some places where brook trout has been introduced, has been stocked, and they are considered non-native in lower Tennessee and in some other basins of mid-Tennessee as well. So what do we do? So one thing is that for each site, because we have a time series, right? We have a time series that spans at least 10 years, at least 4 years of sampling, and all those criteria. So what we can do is that for each site, we can fit a linear regression model to quantify the temporal trend in. We choose 3 things to look at: non-native species richness through time. So for instance, the first diagram kind of exemplifies this, where y-axis is just the number of non-native species for each sampling year. And then the second one is the log response ratio of non-native richness to native richness. So it tells us, the dominance of non-native species. How many non-native species are there in relation to native richness. And the reason why we did this log ratio is because I found that it's a meta-analysis method. By taking a log ratio, it makes the values more symmetrical and it behaves better. So you can see in this case, for number 2, y-axis is your log response ratio of non-native to native richness. And then your x-axis is your year, and you can see that we fit a trend and it's increasing. And your third one is abundance. So it's the same as 2, it's log response ratio, but it's a log response ratio of non-native abundance over native abundance. So again, it tells us something about the numerical abundance dominance of non-native species, non-native fish over native fish. Why do we do 2 and 3? Because we know that, based on our experience of going down to a stream, sometimes we might get, at a given site, at a given time, we might get a lot of individuals. It's just due to luck or just due to weather. Or sometimes you go out with a different crew and you might sample for a little bit longer than you might want, think that you would want to or you plan to. And so abundances is not a good way, abundances tend to fluctuate a lot. So by you doing log response ratio, we are looking at non-native fish richness and abundance, but while controlling for the total number of species that we caught, total number of individuals that we caught. So it helps to control for differences in sampling effort. So after getting these trends, right, we want to predict like what makes some sites have positive non-native species richness trends. What makes some sites have positive non-native dominance trends, richness dominance trends, or what makes some sites have more positive non-native abundance dominance trends. So we want to kind of use, so we use regression models to model these response variables. So these are trends. So we're trying to predict the trends. And we predict the trends as a function of kind of climate-related trends. So we think that, over the past like 30 years, we know that the mean flow has changed in many cases. Some cases they haven't, but in many cases they have. The seasonality of flow, CV means coefficient of variation. So it's a measure of seasonality while controlling for the mean. And then mean temp, so temperature has also changed. So we're talking about the stream temperature that we use from the model that the machine learning model that we fit. We also, for predictor variables, we also use average values across time series. So kind of like, perhaps places that are already more seasonal in their flow, or in places that are, in general, on average, being warmer, they're more likely to have a positive trend in non-native species. So we included those as well. And we included temporal trends in land cover because, in addition to flow, we wanted to look at the effects of land cover as well. So we've got flow, we've got temperature, we've got land cover. And lastly, we included dam density of road crossing, etc. So these variables are there to kind of like test the accessibility hypothesis because, places that have greater dam density within the catchment, it might be more altered, and so it might be more invasible. Places that have higher density of road crossing people might go in there and throw their pet fish that they don't want, or they might go fishing and dump bait fish in there as well. And those are all commonly cited sources of invasive fish. So we added those variables in as well. So what did we find? So we found, the main story here is that, as you see, that there's a lot in here, but these are the y-axis shows you the variables, the predicted variables that are important in predicting for the trend in non-native species richness. So the response variable is non-native species richness, all right? And these variables are, after model selection, these are the variables, predicted variables that correlate with trend in non-native richness in the top model. So they are all important, and you can argue that the ones that show significance, those with the star, so the more stars you have, the more significant it is. The ones with the stars, are even more important. So all these variables are already important because they have come out in the top AIC model. Okay, so what do we see? So interesting thing here is that we see a trend. Let's look at the significant trends first. So for this, there's a trend in mean flow. So it's negative. So the numbers is like 0.000 something. So the code or the program actually rounded it down. So it doesn't really make sense, but it is a negative number and it is pretty significant. So here, it's just trying to say that as the trend of flow, right, in places or at streams where the mean flow is decreasing through the years, there is more non-native species that come in. So that the trend in non-native species richness is increasing when flow is decreasing, all right? Another thing that's interesting here is if you look at non-native species that's defined based on species that's from outside the Tennessee River Basin, you get this CV flow as well. So CV flow is in both models, and CV flow is flow seasonality. So you can think of it as the trend in seasonality through time. So here, as flow becomes less seasonal through time, there are more non-native species richness. Non-native species richness tend to increase through time, right? So that's really interesting because we know that, from being out there in the field and kind of talking to people who know fish and, like CFI who actually try to breed the fish to repatriate them. We know that flow seasonality is very, very important for native fish. And here we found that, in places where flow seasonality decreases, we see an increase in the trend of non-native richness, which is really interesting. And for the next couple of slides, you see the same thing, right? So this is log response ratio of non-native richness to native richness, and see the same thing as CV flow trend, the flow seasonality being really important is negative again. So as flow becomes less seasonal, more of the same through the year, if there's less seasonality, I think what's happening is that it's favoring the non-native species and is impacting the native species. So we see this effect. Here, there's also interesting. We have to talk about crop trend as well for the previous one. So as cropland tends to increase, here we are increasing the number of non-native fish. Over here, there's no crop landing trend. There's a weird, unexpected development trend. We can discuss that, but I wouldn't go into it that much. But here you can see that as negative, as significant as development is tree mean. So this is the amount of tree cover in a given site or a different catchment. And as tree cover for catchments with low tree cover, the trend in non-native richness dominance tend to increase. So for both cases, it's the same thing, right? So, and lastly, we look at dominance in just the number of individuals, not the number of species. So the dominance in the number of non-native individuals over native individuals. You can see again, CV flow is there again, right? Here is non-negative, I think the p-value is 0.07, but for the HUC2 non-native species, it is quite significant. So again, it's kind of consistent when you see that the reduction in flow seasonality through time over the past 30 years, those sites that have reduced flow seasonality are also the sites with an increasing trend in non-native species. Yeah, so here are some of the conclusions from community analysis. As I said, I think flow variability is really important. Decreasing flow variability is a problem because it is associated with increasing non-native richness as well as abundance through time. Temperature is also important in driving some non-native metrics, but again, it's not as consistent as flow variability. Typically, some warmer streams are associated with increasing relative abundance of non-native species, and sites that are warming more quickly have increasing trends of relative abundance as well, but it's only for one metric. And later, when we look at the SDMs, I can offer a potential explanation for this. Land cover change is associated with increasing non-native fish through time. We see a trend of increasing crops associated with increasing trends of non-native richness. If a site has more trees in general, it seems that it helps to resist invasion. So that's interesting. There's an unexpected trend with developed land where increasing trend in developed land actually decrease non-native species. So this is something that it's unexpected and we'll need to consider it a little bit more. There's some indication of biotic resistance through native richness as well, because in the richness model, it came up as potentially important. So just very quickly, I know not much time left. The second part that I want to present is really, here, before this, we look at retrospectively what's happening to the fish communities. Now we look forward to see what climate change may actually mean for non-native fish. So same thing, we identify non-native fish as per before, And we use species distribution modeling. So we use 5 machine learning algorithms, random forest, maxent, artificial neural networks, gradient boosting machine, and extreme gradient boosting to fit ensemble SDMs. And SDMs, they basically relate observed occurrences with different environmental climate variables. So as climate changes, we can then project where species are able to live in the future. So this is a schematic of what is done. This is an example of a sunfish. And these are the input data, flow regimes, temperature, precipitation, land cover, and geomorphology, like the size of the stream, the amount of clay in the stream, that kind of stuff. Because those are variables that are really important for fish. And then we train the model not just based on, as I said, not just based on the Tennessee Cumberland records, but also the whole of the US, because these fish that are native to some of these fish are actually non-native to the US, but because we're using flow data that's only present in the US, we can only use the fish distributions within the US. But to train the data, we're not just using Tennessee data. I just want to be clear that we're using the entire US, right? So after training the data, we can project habitat suitability across 2 RCPs and for 10 GCMs, and we average the likelihoods, and then we do thresholding. So this is the last part that I just want to show you some examples of the results that we have. Like for instance, silver carp. So it's a little bit small, but basically we use a P10 threshold to denote or to identify watersheds where it's suitable for silver carp. So this, like talking to the managers, is this the different species of Eurasian carp and Asian carp seem to be of really high management importance because they seem to be spreading. As you can see, these are the occurrences. The pink ones are occurrences that have been recorded after 2010. The green ones are from 1980 to 2010. So you can see that it's really spreading. So we are using the 1980 to 2010 as contemporary data to kind of fit the model and projecting it forward to 2035 and 2065. You can see that there's an increase in the yellow areas. And you can see that the increase is up the Tennessee River main stem, right? So by calculating the watersheds where the fish is habitat suitable for the fish, you see in the next 10 to 40 years, we see an increase in climate change is expected to increase the suitable range by about 45% under the RCP 4.5 scenario, and under the RCP 8.5 scenario, 59% of suitable, 55% increase in suitable range. For managers too, we can use different thresholding methods. Like for instance, just want to quickly talk about P10 versus P0 or minimum training probability thresholding, where P0 means that it's more permissible. If the managers don't want to take risk and they want to identify as many areas as they think are suitable as well, then we can adjust the thresholding so that we can identify, so that we wouldn't wrongly exclude places where this species would actually occur. All right. So for a lot of invasive species, some people suggested using P1, which is, instead of P 10%, they use P1%, or some people use P0. So this is an example of a fish using both thresholds. You can see that the fish is likely to go up the main stem due to climate change. Another example is brown trout. We know that brown trout is high elevation, all right? So these high elevation areas are going to warm. And as they warm, the range is likely going to decrease because they're going to be out of the Tennessee River Basin. There's nowhere else to go, right? As streams go up, they get cooler, but there's nowhere else to go because the fish can go downstream because it's warmer. So you can see that for brown trout, there's a reduction in range under the 4.5 scenario and under the 8.5 scenario. So this is interesting, right? Because it kind of explains, or at least it confirms what we saw in the community level analysis, where temperature there is some effect, but it's not as big as flow. Because in the Tennessee River Basin, there are fish that are warm water invaders, warm water non-native species like the carps, or there are species that are cool water or cold water like brown trout that are non-native as well. And so they respond differently to temperature. So when temperature increases, we're not going to get similar responses. Like for instance, brown trout is going to probably decline or would have declined in the past 30 years if the temperature is increasing in those sites. But the other warm water invaders would have increased. And so temperature doesn't really, we can't really generalize the effect of temperature, basically. So by stacking together, so this is a map where we stack together, we just fit 7 exotic species, meaning species that is not native and originates from outside the US. So we're talking about the Asian carps, we're talking about brown trout and things like that. And we can identify areas where there are future hotspots for this non-native species. And as you can see, places that are upstream of the main stem seem to be increasing in suitability when we compare the historical to the future scenarios. You can see all these areas are increasing. So it's slightly upstream of the main stem. And those areas are the areas where all these probably the warm water species is going to invade. On the other hand, you see these areas up in the Blue Ridge, right? These are the areas where you get rainbow trout, brown trout, and things like that. They are cold water species. So as it warms, you see that there's a reduction from this brown color to blue color in the future. This reduction is because rainbow trout and brown trout, they're likely going to decline in those areas, as what I showed. So these are the things that we can get from the data. And we have started looking at native species trends as well. And it's really cool stuff. But there was a lot of cleaning and all that to do with this. So yeah, I think I've gone too long, so I would like to end and we can chat about stuff if you have any questions. Thank you so much. So these are the acknowledgements. Really want to acknowledge all my collaborators.